Hello, everyone. I know it's one of the last talks for today. I hope you still have some energy, especially after lunch. So today's talk is going to be more of about like a type of an industry talk, I would say. It's not going to be a lecture. There's not really a way of me giving you a lecture about this. What I'm going to do is I'm going to talk about a real-world application of AI, specifically applications in therapeutics, in molecule design. So let me tell you a little bit about myself first. So basically, I'm a physicist by background. I did my bachelor's here in first best place, so it's Belgrade, in physics. And then I moved to Europe, where I did my master's in statistical physics. It was more applied to complex systems. And after that, I moved to London, where I did my PhD at Imperial College London, where it was the first time where I kind of got introduced to biology and biology problems and machine learning. And so that's where sort of my career path takes a turn, and I started learning more about biology problems. So most of my time I spent, it was actually in academia. It's only the last 3 years that I'm in industry. And this talk is going to be about my sort of experience in industry and how we can use AI to accelerate drug discovery. Right after I finished my PhD, I moved to New York, where I did my postdoc, my fellowship, and I became a research scientist at the Flatiron Institute in New York. That's where I sort of collaborated with a lot of NYU people. And then there was a COVID time, and like everyone else, we were all stuck in our homes basically, and I was pretty bored, and I needed another project. So I decided to try my luck in industry, not by applying to different places, but actually starting a company. So I co-founded a company called Passion Design, and things were going really well. It was most of the stuff and talks to the VCs and a lot of investors and potential clients happen over online, which made things really fast. And everything was going really smoothly, actually. Right after 6 months into the whole thing, the company was acquired by a bigger company called Genentech, which is one of the sort of pioneers in biotechnology. It's a company based in South San Francisco. It was right after 6 months, which was really great. It was right before we signed the seed round, so things saved me a lot of nerves, I guess, a lot of time that I would probably spend to make things work. So right after that, we moved to Genentech. We were fully integrated into their R&D department, and that's where I'm sort of started building the team and focusing on developing AI methods for drug discovery. And since then, I've been working at Genentech. So this lecture is going to be, like I said, more an industry talk. So we'll talk about real-world application of AI in drug discovery. So by this time, you probably covered some concepts in deep learning, advanced architectures, generative AI, maybe a little bit of Bayesian optimization. So you're going to hear all of that, a little bit, like small pieces in this talk, and how it's sort of applied to drug discovery. So this is the outline of my talk. So I don't know actually how much you are familiar with some of the concepts in biology. Actually, how many of you are maybe doing a PhD in computational biology? All right, so there are a few people, okay? Or using some kind of biology data, it's genomics data, protein sequences. All right, cool, nice. So in the first few slides, I'll cover some basic concepts from protein biology and pharma industry. So I'm going to mainly focus on how we use these AI techniques for developing a specific type of therapeutic drug molecules called proteins or antibodies, basically. Then I'll talk a little bit about how AI has a potential to actually transform the whole drug design industry. And then I'll talk about these 3 problems. So we'll see if we're going to have enough time to cover all 3, but I'll talk about affinity prediction. Affinity prediction or affinity binding or binding prediction. So this is the most important part in drug discovery, so ensuring that your molecule binds the target. So one thing I just want to warn you is that, unlike other industries where the data is just easily generated by, addictive people leaving their digital trace on the internet, and then you just scrape the internet for the data and you clean it and you train your beautiful model. In this type of industry, in biotech, so every data point is extremely expensive. So it costs thousands of dollars to actually generate one data point. So that's going to be reflected in the challenges of these problems that I'm going to talk about here. Most of these models, they need to work in a low data regime. They need to take into account a lot of uncertainty. You need to sort of model for uncertainties. You need to take a lot of things into account about this data. So the second problem is this multi-property molecular lead optimization problem. It's pretty important for developing therapeutic molecules that have exact type of properties. The last thing is called de novo design. It's one of the grand challenges in biology, computational biology. If you're in academia and if you solve this problem, it'll get you closer to Nobel Prize. If you are in industry and you solve this problem, make a company around it, it will make you extremely rich. So the thing is that there are already some attempts to do this. Most of it is not exactly what I would like to see. It's pretty much disconnected from the reality of drug discovery, but I'll tell you about our attempts, how we are trying to solve this problem. The last thing is like, how do we couple all of this together in this kind of specific settings where we don't have enough data? So we want to do the experiments coupled with AI, and how do we do that effectively to collect the right amounts of data that are extremely expensive to train or retrain these models and make them better and better. So this thing is called lab in the loop. So that's the technology behind what my company Genentech is doing on AI side. All right, let's start from like very basic concepts in biology. So I apologize to be like, so the first few slides we're going to introduce some basic stuff from biology. So this is the central dogma in molecular biology. Basically, the proteins, the molecules that I'm going to talk about that are actually used for therapeutic purposes are synthesized from DNA. They are composed of these polypeptide chains with like a bunch of amino acids. Amino acids are molecules on its own. There is 20 naturally occurring amino acids in proteins. Here you could see the table of them, and they're usually one letter code that we use to denote each amino acid. Now, if you zoom in into this chain of amino acids, and you look at one particular amino acid, like so each amino acid is a molecule on its own, right? So it has a bunch of atoms, and you can see the basic structure of that amino acid over there with this central carbon atom, C alpha atom in the middle, connected to another carbon atom and oxygens, and then nitrogen and hydrogen. And then you see here the R, which is like a residual side chain group that actually determines the type of an amino acid that you're dealing with. So for example, for phenylalanine is like, NH2. All this time I actually didn't memorize any of these things, so I still don't know the right names of the amino acid, but basically if you put them together in a string, in a sequence, you actually get something you can work with as a machine learning scientist. So it's almost like a sentence, basically, that tells you exactly how this sequence is going to fold up into a 3D structure that is represented over there, and what its function is going to be. So the next step is the function, which is basically what is this protein going to do, where it's going to bind, what it's going to activate. In therapeutics, we care a lot about these functions. So we care about being able to characterize functionally these proteins for a various set of properties, because the therapeutic molecules, the molecules that we use for certain diseases, they need to satisfy certain functions. They need to fall into certain ranges. Now, computationally speaking, going from sequence to structure is you all already know, it's a structure prediction problem. It's the thing that it's been attacked by DeepMind and almost solved. So we have pretty decent methods there that are doing that kind of thing already really well. The second thing is what I spend most of my postdoc on, which is basically going from 3D structure to function. So it's actually now looking at this bunch of atoms that are structured into this kind of we call protein fold. What is the function of this protein? What it's going to do exactly? Now, in pharma or in biotech, we actually don't care much about the problem that AlphaFold was focusing on. We actually care in a reverse problem, which is a problem of I give you a function, you design a molecule that has that function, right? So it's a reverse problem, and that's what I'm going to be focusing on in this talk. Now, what is important here to know, right? You don't need to know biology, you don't need to know these 20 amino acids. Like I said, I didn't memorize them still, and I've been working in this field for some time now. The important thing is that you can represent the protein as a sequence, and you can represent it as a 3D structure. And this is kind of will determine what you want to do with your machine learning models. Now, another thing you need to know is that the sizes of data sets that we have in biology, of course, are nothing like compared to what you can get, you know, if you work in other industries. But these things that are publicly available, one thing you need to know, so there's like these two databases, UniProt and PDB. UniProt is a database of all these protein sequences that are obtained from, you know, a lot of organisms, many sort of, you know, literally covering the entire evolution. So from small bacteria to like big organisms. So you have all these proteins sequenced. So you have all these sequences. And you can see here how the database grows over time. This UniProt KB database has almost 20 million sequences in it. Now, to functionally characterize protein, like I said, you need expensive techniques. So that's why you see the green curve not really growing that much. So this is the number of proteins in UniProt that are functionally characterized. That means you have the labels for these sequences and proteins. And the third thing that you see practically is not growing, but actually it is. It's just, it's a different scale because of the scale on the y-axis. Is the number of these proteins with available crystal structure, like a 3D structure. Getting a 3D structure of a protein is also extremely expensive, unfortunately. So we don't have that available for a lot of things. So this is something you need to take into account when you want to build some models. There's one more thing, which is how this relates to companies and biotech companies. I'll show you about that, like how much of the proprietary data is there that you could also use. I'll show that in the next few slides. So the type of protein we're interested in is this thing we call Y-shaped protein you see on the left side. This is an antibody. It's an important protein, very important. It protects us from the outside world. It blocks it's generated by our immune system, and it blocks all the foreign pathogens like bacteria, viruses, and everything. So this is something that that the human body can generate, and it's exactly what it's used in drug industry a lot. So these antibody-based molecules account for almost 30% of FDA-approved drug molecules in 2022. So these are really important things to design, basically. And if we can use AI to do that, that would be great. These are some of the typical diseases that we hit or that we're trying to cure with these molecules. So one class is these autoimmune diseases. So these are the things like arthritis, Crohn's disease, asthma, etc. Cancer is a big class. And my company, in its portfolio, almost 90% of the things we're designing with AI and with the standard conventional things are actually cancer drugs, right? So, and we're trying to solve that with antibodies. And basically, You have cancerous cells, you want to hit the right receptors on these cells, you want to make the mechanism of killing of these cancerous cells, and that's why you want to design these antibodies, basically. And one of the very first antibody-based molecule that was ever designed was designed by Genentech, and it was used, it's called trastuzumab, and it was used for breast cancer, actually. The last thing is like this Alzheimer's disease, which is also important thing that was actually reported last year. There was a discovery of this antibody called lecanemab, which basically showed like the decline in the Alzheimer's disease in patients, like if you really take it. All right, that's just to show you why is this important and why do we care about these things in biotech industry and where the money is, right? So now taking a closer look at this protein, that this specific type of class of protein that we're designing, these antibodies, this is something that are basic concepts that that's gonna show up in the next few slides. So this thing this big thing you see there, this in pink, it's called a target protein or an antigen. It's a type of a protein that you're trying to hit with your antibody. This thing you see here in green and sort of cyan color with gray as well is an antibody. Now, this is a cartoon type of representation. This is what we use in structural biology to represent the fold and to represent the whole antibody-antigen complex. So the target, we call it an antigen as well, is something that is often implied in some diseases. It could be of various shapes and formats and sizes, basically, and that's the challenge, is that you want to design this thing that is very constrained in a way how it looks and how it behaves, that antibody that binds that target, basically. So this is an example of actually that molecule that I told you that was first designed to cure breast cancer. It's called trastuzumab, and it binds this target called ERBB2 or HER2, which is implied in breast cancer. Now, looking at so like I said, the target can be of various kinds of topologies, formats, sizes, depends on what type of disease, what type of program you're interested in. Usually, in biotech industry, this is something that is given to you. And you need to design this thing here. Actually, you cannot see my cursor. Oops. So this thing that we're trying to design an antibody, right, it has these two chains called heavy and the light chain, and they form this variable region of an antibody. The gray region is called constant region. We are mostly interested in designing things that are in the variable region, that are sort of these two arms that are establishing the contact with an antigen. Mostly, actually, we only care about the tips of these arms, right? So about these things we call CDR loops. This stands for complementary determining regions. And this is the major part of that antibody, of that molecule, where you want to design some mutations so that these things binds better or it has different properties, right? Below, you see the sequence for the heavy chain and the light chain. So that's the amino acid sequence. Like we said in the previous slide, this is what fully determines the 3D structure of the molecule. Now, if you zoom in into the actual structure of the molecule, you'll see a bunch of these atoms there. And you can see there's a lot of degrees of freedom that you need to model with your machine learning model right there. So basically, if you want to, let's say, train. So when I say design, what do you think it's the type of model we're going to use here? It's a generative model, right? Yeah. So you need to train a generative model that learns all this complexity there of all these atoms, of all these orientations, of all these positions, when you're actually designing this thing. And then you need to also design the sequence, right, that folds into that 3D structure. Yes? No. So the antigen in the cancerous cells, the antigen is something that is on the cancer cells. It's like a, I don't know, it could be a receptor protein. And what you're trying to do is like you're trying to design the antibody that hits that part of the cancer cells through that protein, the antigen, and then it activates a mechanism. We call it a pathway. I mean, I cannot teach you all the biology. I also don't know a lot of these things, but and then it kills the cell, basically. Oh, yes. I mean, not only that, but you can run a classifier, and it will tell you if it's going to kill or not. But that's based on a bunch of data that you need to collect first for a bunch of antibodies, and then you need to train which ones are going to kill, like by looking, let's say, at the sequence. And then the sequence could have some mutations here. It could be a variant of the sequence, and then some of these will not kill. So some of it will perform the function, some will not perform the function, some will bind, some will not bind. That's what you saw with AI, basically. So we have this thing called functional assays that we run for a bunch of designs we make, and these things tell us, yes, this achieved what we wanted. That means you can measure the phenotypic change in a tumor tissue or something, and then it tells you, yes, that killed the cell or not, for example. It helps in the disease. There are different mechanisms of action, that's what we call in biotech, by which these antibodies can act. Sometimes it's just blocking. That means maybe this part here of that antigen, it's called an epitope, it usually binds some other protein, and you don't want that to happen because that promotes different mechanisms. And then instead what you do, you design an antibody, a drug, and then that binds that epitope or that region of the antigen, blocks it, and then this other molecule cannot bind that there anymore. And that's the way of actually doing curing disease, basically. Now, there are, like I said, many different mechanisms of action and many different ways of doing things. I'm just trying to introduce here some basic concepts so that we know what we actually need to focus on when we are building the AI models, right? So most of the time, like I said, just one thing to remember here is that there is a sequence of a light chain. There's this 3D structure. The loops are the mediated of the interaction, like this thing called CDR loops. Now, loops, they come from this protein concept that you have actually 3 secondary structures. It's like this alpha helices, this thing that you see as a, I cannot point. Is there any way I can use some? It's okay. Oh, awesome. Sorry. All right, great. So these things are called the loops. Can you hear me? Yeah. And this is usually how the binding happens. It happens by these loops in the antibody side establishing the contact, literally the physics forces that exist between the antibody and the antigen, and that's how you kind of achieve your function here. And so if you look at the region here and you look at the atoms and orientation, it's pretty complex. So if you really want to train a generative model there, it needs to learn these things, right, properly. Now, all right, so how do we train any model? Doesn't matter, like, is it a generative model or we want to predict some function, right? So first we need to see how we can represent this complex structure, right? It's not an image, it's not a text, or it could be, right? It is a text if you just work in the amino acid space. Sometimes that's what you want because amino acid space is always available. Like I showed you the graph of how much data, sequence data is available compared to structured data. It's similar kind of thing even in the companies, right? To generate a 3D structure, which is the most useful thing that you want basically because it directly tells you what the function is. It needs a lot of investment, but sequences are available. So if you can somehow skip that part of generating structure, going directly to the function, that's the best thing you can do. The first thing you could do is what? Like if I tell you, I'm going to give you a bunch of antibodies. I don't know which ones are, or I'm going to give you a bunch of proteins. I don't know which ones are functioning, which are not, which are binding, which are not, right? What would you do? What kind of model would you put together? I give you a sequence, for example. Amino acid sequence, right? Anyone? Yeah, you would do classification and regression, but like, what kind of architecture? What would you use? Yeah. LSTM, transformers, CNNs, yeah, and that's exactly what it's used most of the time. You could also featureize that sequence. You could use chemical descriptors. You could use some evolutionary information. That means you can align that sequence with other sequences in the database and see the variation of each amino acid position in the sequence. So that's what's been used. This last thing is used by deep AlphaFold people, right? The way how they predict this by looking at this is the input to their model, is first they compute this, MSAs, multiple sequence alignments, and then that's how they featureize the sequence, and then they plug that into a extremely complex architecture. Structural features is when you have a structure available. You could do a lot of things. You could voxelize it. You can, well, the most natural thing is what? When you have a cloud of atoms, right? Is to make it as a graph, right? Why would you do a graph versus voxels? Anyone knows any advantages or disadvantages of one representation of the other? Yes, that, and what else? Sorry? I don't hear. Size, yes, you cannot fit into memory every time the voxel representation, yes. Okay, yes, but there is one, the most important. That all is true, by the way, and it really matters when you're building the model and training. Yes? The connectivity, what do you mean by that? You kind of have that in voxels as well, because you know which boxes are close to each other and things like that. So it's captured, maybe at a low resolution, but it's there. It adds to some detail. But like, what is the most important thing? The whole field is like publishing at NeurIPS one paper after another on these things. So it's graph neural networks, yes, but there is one property that these graph neural networks satisfy. Invariance, exactly. Exactly. So what does that mean? When I rotate this molecule, should its physics change? Should its energy change? Should its property change or function? None of these things should change, basically. So technically, you need to think, when you think about representation, you need to think about what the architecture is that you want to have that will respect, formally it's called SE3 equivariance. That means it has to be invariant on the rotation and the translation things, right? So a lot of things, a lot of GNN models, they have that built in, right? I'll tell you, if we have enough time, I'll tell you a little bit of a crazy story about how we did that without using GNNs, maybe when we get to the de novo part. Okay, these are the things that you can use to featureize things. Of course, you can also do the protein surface, so that's a third representation. So you can look at the local topology of this molecule, etc. Right? You could also learn the features, right, by training some kind of models. The typical things are done like with language models, right? We train language models on proteins, right? On protein sequences, there is quite a number of them. ESM is one of the famous one by Facebook people. It's trained on all of these millions and billions of sequences available in publicly available databases. So this is something that, it's like all the evolutionary data you can use for training these models. There are those that are specific to antibodies. It's called Antiberti is one of them, which is just trained on antibody sequences. There are some older ones as well, using some NLP tricks as well to train these models. Structure features, there's also that. Now that we live in a world where next token prediction can solve every single problem, everything can be tokenized, so even the structure. And so there are papers that are actually tokenizing the structure using various tricks. And then you can train these kind of multimodal language models where you plug in the sequence and you plug in the tokenized structure and then you tokenize sequence, tokenized structure, you have a combination of tokens and you train a model basically. So you can do all of these tricks. The representation is important for what? for all of these downstream tasks that I told you about, and I'll be covering that more, which is predicting function, predicting properties, etc. This is just a visual of some of this representation stuff, right? So the protein graph, and then some of the architecture that usually go with that kind of representation. The backbone frames, this goes back to that thing that I was explaining, how the residue is a molecule. There are certain bonds between atoms, and you can form these frames, and then you can define the position of each frame through rotation and translation, and that's what is used by AlphaFold and Rosetta Fold people. Sequence, one-hot encoding, that's the most typical thing. Voxels, so we covered all of these things. This distance space stuff, these are very cool when you really don't want to struggle with equivariance and any of these things. This is another sort of historic way of showing this, or how this is sort of used. Sometimes this is this kind of classification that exists in the literature, which is 1D. It's just a sequence of physics descriptors of the sequence, basically. 2D representation is a contact map, so which atoms is in contact with which, graphs, surfaces, point clouds, we cover that, except for this last thing we call the 4D representation, which is, you learned that the protein can be a sequence, just a string of letters. We learned that it could be a 3D structure where you can look at the atoms, composition, the distances. And the thing is that these things move as well. They are not static. That makes things much, much more harder. So they actually move because there is a physics of interactions between different atoms, and especially if you do this in a specific environments, there's like forces between these atoms. There's a lot of stuff. All right. All right. Okay. Taking a step back, let's talk about machine learning now. Hopefully we introduce all basic concepts now. So designed, we said you might be using generative AI, right, to come up with a sequence. What's the key challenge there, right? If you don't have an AI, right, how would you design a sequence? I told you there are 20 amino acids. You put them together next to each other, it forms the sequence chain. That chain folds, gets your protein, it does the function. If I were to design this thing to have a specific function, that means if I put a specific combination of amino acids, and then I have the function that tells me that's the function, right, of the protein, that's the fitness, that's the stability, right? The goal of finding the right function, finding the right sequence that perform that function is actually looking at what kind of combination of amino acid I need to make that will have this function. So how big is that combinatorial space? Does anyone know? For a protein of length L, 20 to the power of L. It's a lot. So crazy people in pharma were doing this thing in that way. So it was literally doing that, literally making combinations, or they call library design. Library is like when you really start and putting random letters and trying to see which of these things are going to express which of these things we're going to have function, etc. Another thing we're very interested in is not starting from zero. Starting from zero will be close to de novo, but we sometimes have a sequence, protein sequence, and has a problem. So now you need to make a variant of that sequence that will have improved function. So that means you need to introduce some mutations here. Even then, it's a combinatorial problem. Where are you going to introduce these mutations? What you're going to mutate to what? If you're especially designing some regions, like the CDR loops, for example, etc. Even looking at the smaller region of the protein is still a huge combinatorial space. So it's not possible. But like I said, in pharma industry, people do this, and it's called mutagenesis, point mutations. They measure each of these things, and then they tell you this. But of course, when they go and combine stuff, it explodes very quickly, and you cannot do anything there. The two major approaches in drug design are these two: rational design. This is like a creator of Rosetta, the person who did the first physics-based molecular modeling stuff. And the idea is that you have a structure, you have a backbone structure, and you need to come up with a sequence that minimizes the energy. And in his algorithm, that's done completely randomly. You make substitutions, changes, etc. In the directed evolution, which is Francis Arnold's work, which is a Nobel Prize work, is you start with some variant pool, you start with your parent sequence, you make some variation of that sequence, you have a functional screening, you select only the good ones, and then you mutate them again, and then you repeat the cycle multiple times until you converge to something that looks like good. But again, introducing these changes there is completely random. So it's a very difficult problem. So how do we approach this problem, right? Now, we know that sequences and structures are high-dimensional data. There is this concept of data manifold, where this high-dimensional data can be actually projected in a low-dimensional space called manifold. And with that manifold, things behave nicely. That means you don't have many options. In the combinatorial space, you have many options. You can take out your input sequence, you can mutate it this way, you can this way, and then you can just have so many combinations. When you are on the manifold, and let's assume you train the model, you learn the manifold, you can actually move in a very restricted directions to still preserve or maintain the identity of your data points. So there are only a very few number of ways in which you can move. If you train an image manifold, right, MNIST, there's only a very limited way in that space in which you can move so that you still generate a number 4 digit. Similar thing with proteins. So if we can learn this for proteins, then we can have this whole optimization problem instead of original combinatorial space, which is very hard. Instead, you do it in a manifold space. And sorry, this is something that is probably displayed wrongly. But anyway, you can define your optimization function optimizing over X that is actually alignment on the manifold space. There are many ways to do this. In early days when we were like doing the startup, we used something that was, at that time was not, it's close to diffusion. That was way before diffusion models. But it's basically called denoising autoencoders, right? Which is a way of training your model by taking an input, corrupting your input, and then learning a function that can actually denoise it or remove that corruption. In that whole process, of injecting that noise, so what you're doing, you're mapping your noise distribution to the data distribution process, and basically you are, I apologize for this is probably just a PowerPoint thing. So by doing that kind of thing, by training some kind of an encoder and decoder, in this process, you are actually able to sample the manifold, right? That means if I'm standing on a manifold, I take a data point there, I introduce some noise, and then I run a step of encoder and decoder that puts me back on the manifold in a different point, and I generate another point that actually is on the manifold. So this is how you could actually sample stuff. It's almost like a one-step diffusion. Nowadays, diffusion models, they use like a scheduler and all of these things to learn even a better representation. The architecture we had at that time was this transformer-based, the most powerful thing at that time that you use on sequences, encoder-decoder thing. But one cool thing that it had as a component there was this classifier or this function predictor that was learning also how to map from that manifold to the function. And then if you have that, if someone gives you a function, you can actually use the gradient from this classifier and guide the sampling in the direction of that gradient on the manifold. So that means you don't move randomly on the manifold conserved space, but you actually move in the direction of that gradient that tells you this is going to maximize the function, right? And so that was another block here that was built into the system that we train on a bunch of, basically billions of proteins and all these proteins that have some kind of functional characteristics. Now there are a bunch of methods that you could use to learn this manifold, to learn the data distribution of sequences, just if we're in a sequence space. This is sort of the chronological order of the things that are used in protein sequences, for protein sequences specifically. Of course, there's a lot of things in NLP that exist. The first things, even before AI, ML stuff, this is something, pretty much 30, 40 years old. It's HMMs. This is something you learn, you're giving a set of sequences, you're learning a specific distribution on a specific location on that sequence, right? The frequency of that letter in your multiple sequence alignment. If you want to take some more pairwise dependencies, you can train a POTS model. And then there's an era of deep learning methods where you can learn even higher order connections, and that's basically latent variable models. You could check out all these papers. Basically, these are all attempts to, pretty successful ones actually, to train these models on sequences and to learn the distribution and then to be able to sample new designs from this distribution. There are, of course, in the last 1 or 2 years, a lot of methods that use structure as well, that are actually training these generative models, learning all these complex atom clouds and things like that. And I'll talk about that as well. So let me just check the time. How much time I have left? Okay. Okay. All right. Now, these are some of the early attempts to use, well, not early, but pretty recent actually, but the things that still I have problems with. Let's put it like that, right? These are all Nature Methods papers. They are all trained language models that use, that kind of learn really good sequence representation. And once you, or your manifold, right? And then you can use it for design, right? The first paper is actually, if you remember the directed evolution cycle, the thing of making random mutations, screening, diversifying again, and then screening again and selecting, right? There is a paper that actually, for the first time, replaced that screening stage with the machine learning. And so instead of doing very expensive biologically expensive screening, you actually can run a machine learning model. The second thing was, instead of going again in a random space, you could train a model. It's a UniRep, like a language model trained on UniProt data, bunch of sequences. But the problem there was that these guys who did this, they still were designing things in the original space. So they had a manifold, they used it for training a classifier for just screening, but they didn't use it for design at all, right? Design, they did still original space where they take a sequence and they say, I'm going to mutate it here, and then I'm going to put it into this language-based model classifier or function predictor that is trained on top of a language model, basically, to tell me if this is a good sequence or not, right? So in the previous slides that I showed you, we actually used gradients from the function classifiers to actually guide the design and selection. So that was kind of an improvement over these existing works. Now, I told you that there is some problem with this publicly available data, and this is the reality, unfortunately. So if you want to solve some of these big problems that biotech companies care about, which is designing these molecules, you actually, empirically, you actually cannot use this publicly available data, because the sequences and proteins that are in here are not absolutely related to the type of molecules that you're trying to design. So this here, let's say, represent the all protein space from these publicly available databases. Inside of that space of proteins, which could be all sorts of proteins, right? Inside you have a subclass of antibodies, and you can get them from this publicly available database called observable antibody space, if you want to play with it and train some models. The thing is that things that I work on in the company are right here. And I only have a few clones or a few variants of my molecule that I'm trying to optimize, and it's almost out of distribution compared to the publicly available data stuff. So what can you do with these things, right? Well, there's nothing much you can do. What you could do is bridge this gap by doing additional experiments and doing additional sequencing. And that's exactly what we do in the company to generate more data for these things, because you won't be able to kind of train anything meaningful here. So how do we generate these things? Well, it turns out that the way, if we go back now again to antibodies, the way how antibodies are generated is, again, through DNA, just like any other protein. But there is this whole process called VDJ recombination, which is basically there are 3 segments of genes in our DNA, and they get recombined to synthesize an antibody. And then that antibody goes through a process called somatic hypermutation, which is, again, another biological thing which tells you that this antibody needs to introduce some mutations to bind the target even better. Using this process, you can actually, by torturing animals, you can actually get your sequences. And that's exactly what is done in biopharma companies, right? So if you want to have more antibodies to train your models, what you do is you take a target of interest, the thing that is in human, a cancerous thing, and you inject it into an animal. And then animal develops an immune response to it and starts generating antibodies. So every animal here has a small generative model inside and starts making your antibodies, basically. And the collection of these antibodies is huge. We call it a repertoire. It's of the order of 10 to the 9 sequences that you can extract by sequencing, and then you can send it to some experimental screening. It was the question about how do we know which things work, right? Because we design specific kind of apparatus or functional stuff, right? That will screen for antibodies and that will do things for functions that are done in the right way, right? So you know exactly out of these 10 to the 9 antibodies, which ones are the ones that perform the function, right? And that's how you get the antibodies, right? The initial set, or at least the lead antibodies here. So this is a usual step. It's called an immunization. Animal immunization campaigns, and a lot of biotech companies have actually animal facilities to do this. And to extract these molecules that when you start optimizing them, they can eventually become drug molecules. And these are all the steps that you need to do when you design a molecule. So from that target identification, what is important in cancer, technically we get the target as, it's just a list of things that the target is already known, even though there's a whole research happening right here to tell you what's the mechanism exactly for that disease. Right after that, you do lead antibody discovery, which is like this immunization thing, and then you do all sorts of optimizations. This first step is called a preclinical study. It costs hundreds of millions of dollars, and it takes 3 to 5 years. If you survive here, then you go into clinical stages, and then you can actually test it in vivo. Again, there's animal campaigns. There is a lot of different ways to test for toxicity, test for other things, and then if you reach here, you're done. Now, a lot of programs, a lot of targets, a lot of sequences, they don't get to this stage. They don't even get to this first stage at all. They get killed somewhere in between because you didn't select the right thing. And then I'll tell you why and how the selection is done, but you get killed here. And based on this success of how much you go from here to here, that depends your stock of your company is going to go up or down, and how much is the worth of your company. Basically, will depend exactly on this. But the whole process is 3 to 5 years plus 5 to 7 years, and you can see the prices of things. So what I'm doing in the company is I'm trying to use AI for this first step here, right? For this lead antibody discovery. So use generative models, do the screening evaluation, AI models. Do all of this preparation and optimization as much as you can with AI, basically to speed up this whole process here. Speed up and, of course, reduce the animal consumption and, generally transform this whole process there. The problem is that a lot of things like I told you, gets killed in between in the process, and the reason is because you can even achieve this binding affinity and things can bind to the target. But there is a lot of other properties and functions of your molecule that needs to be in the right range to be considered therapeutics. And that's the whole thing about multi-objective optimization I'll tell you about. Why things get killed and why some of these projects are dead, even though you first couple of years invested a lot of money, is because from the huge number of antibodies you start from, you select really wrong clones from the beginning if you do this in a standard way. So these are some of the ways how generative and discriminative models can help. So we can actually generate better proposals. We can do sequence multi-property optimization. We can do de novo design. If you do de novo design, you actually can replace the whole immunization campaign with AI. All right, I'm going to skip this part. Affinity binding prediction is a classification or a regression problem. The only problem is that you don't have much data there. These are the two things that we use in the company to get the data. But if you think about what the problem is, from the biology perspective, there are these loops, there is a lot of things, there is a lot of dynamics that affects the binding, and you're not taking into account all the physics by your AI systems, and you cannot really model this. That's the first challenge. The second challenge is if you look at strictly from the mathematical point of view or machine learning point of view, which is that this whole sequence space is extremely sparsely functional. Like the whole landscape looks like this. And what does that mean? It means that I could have a sequence of an antibody, and it binds the target, it does the right function. And I just make one mutation, and it can kill the binding. Sometimes I can make 5 mutations, and nothing happens. Sometimes I can make 10 mutations, nothing happens. Sometimes I make 2 mutations, it kills binding. So the function is not smooth at all, and you need to learn it with your AI. So it's a very tough problem, especially in a low data regime where there's a lot of uncertainty and a lot of problems. Of course, what do you do then? How do you smooth the space, right? We talked about that. You go to the manifold space. So a lot of things we do is basically training on top of some language models, and of course we use proprietary data, internal company data to train these models like everyone else. We don't use language models designed by some other companies. We need to train it on our own data to bridge that gap that I was telling you about. Now, binding, and even if you solve the first problem and things bind, you still have a lot of other things you need to optimize for. Now, I don't want to go again. There's a lot of technical domain knowledge stuff, like what this antibody needs to have. Technically, these are all labels or values. Whether you think of it as classification or regression doesn't matter. The only problem is that you sometimes don't have enough data. Sometimes you don't have enough diverse data. But these are all the properties that we're optimizing antibodies for. And all of these things, they need to be in the right range. That means it needs to be stable. It needs to be very specific and binds only the target, but not the rest of the things on the cell and other stuff that will cause a lot of side effects. It needs to strongly bind your target. It needs to express really well so that you can do additional experiments on it. It should not trigger your immune system when you inject it into human. Your human should not reject it, basically. It should not reject that molecule. So that's called immunogenicity, et cetera, et cetera. There's a lot of things. So when you start with your candidate designs and you screen them for all of these things, most of the time you're not left with anything. You don't have a molecule. So if you actually train the models to predict all of this stuff, then you could actually, in that screening, in that early stage, select exactly for the things that you want. And then most likely you won't have problems on the way, and you can save a lot of money. So how do we do this? We couple these things. So we have a thing we call lab in the loop, where we use active learning to improve our models. The models are generative models that have seen a lot of these repertoire sequence data that are already available in the company. So we train a bunch of these models. Then we send the designs, which is millions of candidates or billions of candidates, through this thing called probabilistic predictors that are all models separate for training for all of these properties and functions I told you about. And then we do selection based on their predictions. How is this selection done? It's done to do the multi-objective function. And then whatever survives the screening goes to the wet lab. Once the wet lab does characterization, it goes back to us. We then put it back into the models. Our models get improved. Now, how do you select things that will improve your models? I don't know if you covered that in any of the lectures so far, but that's the key thing, right? This is the person who was supposed to be here and give a talk. It's one of my co-founders and collaborators, and he likes to say that the number of the antibodies we have are right here. The therapeutic ones are right here. This X and Y is your developability space, is like these properties. So we need to go from here and push them into that region of developable antibodies, the things that can be potentially therapeutics. So the problem there is that all of these things that I talked about, all of these properties, are competing properties. That means that if I have a generative model and propose mutation here, it can improve affinity, if that's what I'm trying to optimize. It will immediately make these other properties worse. And then it will reduce the stability. And then I focus on stability, and then I fix mutations there, and then I lose affinity. So that's a big challenge there. And in drug discovery right now, it's done in a sequential way. That means you screen for first thing, and then you screen for second, and then it's a terrible way. The machine learning way is doing a multi-objective optimization and staying on something called Pareto frontier. So what does that mean? It means that I need to stay in the region where I can only make one property better without a cost of reducing the values on this other property. And so that's the only way I can, from my generative model, select things that are good. So if you think about it, what is it? What is that? Does anyone know the technical term in machine learning for doing that kind of optimization? Yes, it could be that, yes. But it's we call it like, it's you know, we call it black box optimization because you have multiple functions that you don't know how you got. But basically you train these models. And then the question is, if you do like a Bayesian optimization, what is your acquisition function? What is the thing that you're trying to do, like what's the metric there? And in Prescient, in my company, we use this thing called expected hypervolume improvement. It's a very intuitive thing. If you're doing this multi-objective optimization, it's just look at your space of samples and what do they cover. And if you have an objective 1 and objective 2, you look at the area that are spanning by this set of molecules here. You're looking which molecules will improve that area. You're trying to maximize that hypervolume improvement change. That's how you kind of do this multi-objective optimization. And you use this property predictor pseudo-oracles to get these values on the objective 1 and objective 2 for some of the samples. So when we do lab in the loop, an iterative process, what we do is that first set of clones, let's say, could be this orange. Now we're doing another set of optimization. It means we select the clones only that improve hypervolume improvement. You can introduce a lot of stuff as well. There's like some papers here you can read about. Probably I provided links there. But the idea is you can account for hierarchy between properties. That means if the antibody has this property, automatically needs to have this other property, et cetera, right? So, but that's the whole idea, is that in one iteration I-1 of our lab in the loop, and iteration I, we compare the hypervolume hypervolume, and then we only design things that improve the hypervolume. That's what we send to the model in the second iteration, and that's what iteratively improves the model, right? I will skip this. Yes, I mean, just to tell you, these are like the two major properties we're doing for, which is yield, which is related to expression of an antibody, and the other one is how tightly it binds the target. You can now predict these two things computationally. So you can actually run everything in silico for screening for the things that improve hypervolume, and then you send that to the lab. This is the result. It's amazing. We were running like 6-7 rounds. These are like multiple projects, multiple targets, multiple antibodies. But what you see is that over time, by experimentally measuring, is that things get better in terms of expression, things get better with affinity, and we're actually designing way better molecules by putting all these things in one loop. And a lot of our classifiers and a lot of things get better and better over time with the number of lab in loop iterations as you're doing this active learning process. Last thing, 10 minutes, I have to tell you about this grand challenge thing, the Nobel Prize level thing, is exactly what I already told you, kind of, which is right now the way we discover the initial set of antibodies is by doing this immunization. And then we can have a lot of multi-objective optimization with machine learning to get to the right molecule instead of making all these mistakes that big pharma does by doing sequential selection and all of these things. But the whole goal, that's my focus in life, is that I want to have only a target and I design a molecule from scratch. Someone gives me a target and a location on the target, like a location on the molecule, and I design an antibody from scratch. I don't need to do immunization, I don't need to do anything. So how do you do this? What's the most popular generative model now? Anyone knows? Diffusion. But what do you need to do to make diffusion generate an antibody that binds exactly that space? You do the 3D, you do all of these things, but you need to do conditional generative modeling so that you make sure that it actually fulfills all of these conditions. There's quite a lot of work on that already. The only thing is that none of this has anything to do with therapeutic stuff, because with therapeutic it's much more harder. You still need to incorporate a bunch of these constraints, a bunch of these objective functions when you're designing a de novo antibody. Many people just focus on affinity, and then they design something that cannot be used as a drug molecule at all. So diffusion, you're I don't know if someone covered this. Matt, could someone cover the fusion? Okay. On proteins, very easy. You destroy protein, adding noise, all the atoms. You train a model that reverses that process, and so you can sample, or you can do some crazy videos like this. Okay, that doesn't work. That just shows like how you basically start from like a completely random point cloud and you get a fully formed antibody. This is the conditional process. I wish this movie can work. No, it doesn't. All right. No, unfortunately, no. This is one of the targets for which we designed the things. We had a location, and you could see really, like starting from a completely random point cloud, like a gas of atoms, how they become formed and bind exactly the location on that target. From some of our models. Last thing I want to say, so going into the specific of these models, right, we call this thing a AB diffuser. You can read more, there's a paper published on it, but we talk about SE3 equivariance being very important, right? And the way you achieve it by having GNNs, right, the message flows in the way, like over the distances only, or in a way that when you rotate the molecule and you do all of these things, you don't lose the equivariance, so your model knows the right function. One thing we did was inventing a completely novel method that actually does this equivariance in a little bit different way. And this is done by one of my, Andreas Lucas, who's in my team basically, who's a GNN person, but for some reason he got crazy and he said, I don't want to do GNNs anymore. What I'm going to do is something very crazy, which is I'm going to represent a protein as a table. And each row here will be one amino acid. Each column here will be coordinates of your atoms, like carbon atoms, nitrogen, all of these things. And it could be like also sequence identity, like which residue type it is. And that's my representation of a protein. We talked about a lot about representations that I was kind of covering because I wanted to tell you about this cool result here. So, and then he said, I'm going to train just MLP layers on this. And I'm not going to care about equivariance at all. And that was pretty, I mean, that kind of architecture we were putting there together, it's called an MLP mixer, I'll get to that. But basically, the way it solves the equivariance is by doing this thing called frame averaging, which basically just sums over the elements of the group, the maximum things, and all you need to do, basically, you run the model 4 times on the principal components, and you sum it, and you solve the equivariance problem. But as a reward that you get is you actually have extremely powerful, highly expressive model with only MLP layers, and you're probably familiar with MLP mixer. You just run MLP layers on the rows and the columns of your table. And you can actually create high capacity architectures in this way by just doing this trick of frame averaging to handle the equivariance thing. So that was the building block of this diffusion model that we were like sort of putting together. And it actually also has some experimental results here. We submitted to the lab. We were able to generate a bunch of novel CDR loops that all had really good affinity values. I do want to stop here, probably. This is like just showcasing like some sort of examples, but I can take some questions now. Thank you, everyone, for listening. Okay. Yeah, the first question was, I repeat the question. The first question was about OOD, like auto distribution, and how we handle that, because a lot of molecules we have, and that's true, I was showing this, is way far away from a lot of publicly available proteins, right? And there is no better way to handle this thing than working in a company that has a lot of money that can actually just generate more data. So that's the way how we handle these things. We just generate more data that bridge that gap and get you more support or coverage around the sequence or structure space that you... With the active learning, yes, that's the part. Because you can expand that, starting from even, like you saw the performances when I was showing, in the beginning it was terrible. It was like 30% expression, and then very quickly in the second or third iteration it became very, very good. Because for that specific thing we started, that seed molecule, it was like 20 data points, 30 data points, and then you start building and building over time, and this is how. Second question is, do we design entire antibody or just the region? Actually, it's just the region, it's just these loops, the variable region, and that is actually pretty good because that fixed region that is not doing the binding, it needs to be human. In order for, like when you inject it in human, so that your organism doesn't detect it as a foreign molecule. And so we use humanized frameworks and we design variable regions. So that's the strategy. Yes. I did not hear the beginning. Was it like, do we... yes. So, is there any resource that shows that data augmentation helps in these situations? Yes, it does. I don't know what specific example, like there are many examples we're doing, but one thing is that there are like a bunch of these molecular modeling tools for proteins that exist out there. They work really well. They've been something that people were developing for 50 years, and then DeepMind people show up, and they design this thing that works really fast. So, but these tools, they can still model pretty accurately the physics. And so many times when we want to design, or we want to introduce some mutations, or we want to fill out that space of all possible sequences that will still function and fold, we use Rosetta. This is molecular modeling suite that is actually just able to tell you this thing will probably not destroy anything. So there's a lot of priors built in these tools that are respecting physics, and then you use them to sample a bunch of synthetic data, and then you use that synthetic data to augment the training of your models. Plus, I told you the dynamics part. Dynamics is also handled by a lot of molecular modeling tools. This is just things that simulate physics. And so if you want more structures or more confirmations of that structure, then you run that tool. It's expensive. It takes days and days to get trajectories of that one molecule. And then you use that data to augment the training. So there's various sorts of tricks we're doing to solve that problem. But augmentation usually helps in all these problems. Yes. All that money that goes from IND to FDA approval, which is billions of dollars. Yes. So it seems like to me, even if you solve the approach design problem perfectly, you solve this massive bottleneck to actually move therapeutics forward. So my question to you is anyone working on modeling for the second half of understanding, like how does the drug distribute in our body? How do we excrete it? How do we metabolise it? Very little with AI that I know of. The question is, when I was showing the typical drug discovery process, the stages, there were like two major stages, which is one is preclinical stage, where you do all this research, and that's sort of where my department is in the company. We only do research. And then there is a whole clinical stage. The first one is like millions of dollars, the other one is billions of dollars. And then the question was there any, and I was telling you I'm focusing on this first part of like getting that lead molecule that checks all the boxes and has all the properties right now, all good, and then it goes into clinical trials, after which I don't have any control. But the question was, is there anyone doing machine learning on that second part? Makes sense to do that, right? I don't know much of the work, but step by step, I would say, I'll do that as well. The whole thing that the Kyung-Yang Cho, who didn't show up at this thing, is that he likes to say, I want to backpropagate he uses this backpropagation thing. I'm going to backpropagate from patient all the way to the atom positions, right? So technically, you do want to put this end-to-end whole process, just like we did with lab in the loop, that goes from clinical all the way to the individual residues, amino acid residues and atoms. It would be really nice. But unfortunately, I don't know that because that data is sensitive, a lot of patient data, a lot of stuff. I honestly don't work on that, but it makes sense to see how much AI can help there. Probably there are some works in the company. I don't know many, honestly. Yeah, but it's a good question, I think. I think it's step by step. We'll get there, I think. Yeah.
Okay. Yeah. We're going to have to stop with questions over here. Can we thank the speaker once again?